In the case of gene pool, it goes beyond a hypothesis. It's actually the data. It's actually how evolution happened. So I just think that that's really cool that we have the ability to extract these genetic data over time and actually visualize how those divergences occurred, how those different species evolved over time. Perhaps it's just the geek in me, but I'm so fascinated by that. And I love that ability of being able to visualize simulations over time.
The idea of code as a living thing, creating out of pure code a universe, a world where things are able to come to life.
This beautiful self-organization and replication, and even the mysteries of how the first cells began.
How did the world come to have so much biodiversity? What is it about our planetary ecosystem that allows so many species to coexist at the same time in the same common environments over long periods of time? I was exploring genetic algorithms as a way to simulate the evolution of morphology and motion control. Now think of all the ways that animals move around in the world, animals of all sorts, undersea, on land. Is there a way to model the evolutionary dynamics that allows these strategies for locomotion to emerge? I decided to put hundreds of these virtual creatures in the same environment, in a common environment, and let evolution unfold naturally. The use of genetics or modeling genetics in virtual creatures was still a new thing at the time in the mid-90s in terms of applying it to computer games. But this is something that I proposed at Rocket Science Games soon after I started working there, and that's where I met Brian.
Yeah, I came in '95. We kind of had a little talk about what I wanted to be doing and what the opportunities were. And they said, well, there's this guy, Jeffrey, he's doing some prototype stuff. We did manage to make a game out of it. A lot of people liked it, but from a marketing product point of view, they were a little bit befuddled. How do you win a simulation? Well, I got this cool thing here. Look at what it does. So that's not necessarily a winning condition.
So I work as a conservation biologist at the Charles Darwin Foundation. My official title is ecologist and biostatistician. And so I live and work in the Galapagos Islands. In my mind, I think this project started back when I was 12 or 11 when I first started playing around with Darwin Pond. This downloadable program that I could play around with on my Windows 98. I would run my computer, let my computer run Darwin Pond overnight and let it run as long as possible to see what would happen and see if I could eventually evolve some master swimmer that was always better than the rest. Then I realized when I started learning about how you could actually copy and paste the swimbots and see the DNA code. Then with really no understanding of evolutionary biology, I had this feeling that there's gotta be some way to compare different swimbots and be able to see how they're evolving by looking at their DNA. I created this little program to kind of visualize the differences in the DNA between the different swimbots that I had run in different simulations. It was enough to kind of start to visualize those differences and get really excited about evolutionary biology. Didn't really know it at the time. And also about the potential for something like Darwin Pond and eventually what became Gene Pool.
So a swimbot is a two-dimensional creature, and it's made out of parts of different shapes and sizes and colors. And these parts are constantly moving. They're constantly rotating against each other using sine wave functions. And these sine waves have different frequencies and amplitudes and phases. There's a huge variety of body types and motions, so lots of potential for many different swimming strategies that could come out of this. So think of the ways that you can paddle a canoe. You can push the paddles back to move forward. You can push the paddles forward to move back, or you can even rotate the canoe. So these are some of the basic principles of physics. Now, these genes determine lots of aspects of a swimbot, not only the shapes of the parts, but their colors, how these parts are connected together, the way they move, and so on. The simulation starts with hundreds of these swimbots distributed in the pool, and their genes are initialized randomly. Now, most of these swimbots will just flail about uncontrollably without any coordination, and they won't be able to get to what they want to get to, which are these little green food bits. Now, these green food bits provide energy, and these swimbots are burning off energy, especially when they're moving really fast. Every swimbot has a heading, and so it can tell the difference between its heading and the direction to a food bit. And that angular difference is a part of its sensory system. Swimbots live very simple lives. They're either interested in food, or if they're not hungry, they're interested in sex. And here's the fun part. Some of these swimbots, initialized randomly, will be lucky. They'll be lucky enough to actually, as they're randomly flailing about, get towards the food where they're close enough to eat it and get energy. And then using those same flailing motions of their limbs, they'll be able to get to a mate. And in doing so, they produce an offspring. That offspring then inherits those lucky genes, and so it goes. The spread of genetics is correlated with the ability to reach a goal.
Let's look at how gene pool wasn't really allowing for coexistence. You seed this population and you let it run, and it's really survival for the fittest. It actually allows for this really intense competition to occur that evolves what we call these little eel swimbots, just kind of one little strand that kind of swims along. So that in and of itself is a really interesting lesson in hydrodynamics. But the thing is, those eels are so efficient that as soon as one of those eels evolves, it very quickly takes over the entire pond and everything else goes extinct. If you're an organism A and you're trying to seek out the food that A's like to eat, you're going to be competing more with other fellow A's for that food than you are going to be competing with organism B, which needs a totally different food. Another way to think of that is they have different niches. We implemented a few tweaks to the code in order to prevent the ability for one of these eel bots to evolve. But of course, that wasn't enough. So the other major change that was implemented was creating 2 different food bit types. There's also genes now that code for a swimbot's preference for a certain food color, and then also for how they digest that food color. Previously, all swimbots could always interbreed with each other. So at least based on the biological species concept, in which species are defined based on which organisms can mate and produce viable offspring, there wasn't the ability for swimbots to speciate. So you have 2 different populations, and then they come together, and because speciation isn't a concept, they can all interbreed. We made this really cool discovery. There's, I think, 250 genes in total, but it was something like only 100 of those genes coded for the way that swimbots move and all of that. But a whole bunch of these genes were essentially junk genes. But the really cool thing is that all of these genes still mutated. There's actually a natural genetic divergence that occurs is just by chance alone, just by the fact that mutations are random, you could take an organism from each group and by looking just at the junk DNA, you'd be able to easily separate them out into the two different groups that they came from. If the junk DNA between two different swimbots surpasses a certain threshold in dissimilarity, then those two swimbots cannot have offspring anymore, and they are considered distinct species. What if we could plot and visualize that evolutionary divergence between these different species over time? This kind of connects back to my original goal and dream of trying to somehow visualize the evolution of different swimbots over time. And now, having a little bit more background in evolutionary biology, I've gotten a little bit better at understanding how genetic data are used to create what are known as phylogenetic trees. We can't go back in time and get DNA from the dinosaurs, from all these hundreds of thousands, millions of species that have gone extinct. And the really cool thing about how gene pool works, and by its very nature, is that we have all of this evolutionary data over time.
I know from my research that human beings have many genes in common with something as weird as a little nematode worm. We have like 60 or 70% of our genes are the same. And that's mind-boggling to me.
I know what that creature is doing. I know what it's trying to do. There's even a sense of sentience, like they're trying to do something. And we project that, of course, from our own experience of having bodies and trying to get here or there. But I think that metaphor, that connection, it's not a bad thing to be seeing and thinking. Because that metaphor, that connection with these simple creatures allows us to identify with them and maybe grok this unfolding drama of evolution in a deeper way, in a more personal way.
It boils down a very complicated, messy world of variables into a system that you can understand and enjoy watching. It's fun. I think that's the main thing about it, is that it's the little wiggling things. Once the student groks what they're seeing and what it means, they understand, each one of these things is different. They can mate and create offspring. We're all part of the universe. And in a way, we're just the universe learning more about itself. These little things that kind of come together, wiggling around. And that's how I feel. Like we're all just little wiggling bots in a way, trying to make some kind of connection with somebody. And it's that connection that is, to me, the beautiful part of being alive.
The eventual goal with Gene Pool Laboratory is to have an entire interface or an entire laboratory where you can go in and create hypotheses, run experiments through Gene Pool, and then extract the data from Gene Pool, upload them into Gene Pool Laboratory, begin doing analyses with those genetic data with all the tools that you need essentially as an evolutionary biologist and ecologist. My sincere hope is that eventually Gene Pool Laboratory can get to a place where teachers can use it to present curricula on evolutionary biology and ecology. Gene Pool gives us that simplified way of seeing these really complex processes that you can't see otherwise as easily when you just walk out into nature. I think that's the fuel to then go out and explore your environment, learn more about it. Maybe even go as far as what I've done, and now I'm working in the Galapagos trying to conserve species. That's the way I see it.